Our next session is Tackling Neglected Diseases, Indian Healthcare Breakthroughs. We invite on stage Anand Ranganathan, professor at Jawaharlal Nehru University, author and columnist. Shailja Singh, professor, Special Center for Molecular Medicine, Jawaharlal Nehru University. Soumya Pati, Associate Professor at Shiv Nadar University, Delhi NCR. They will present their papers on molecular medicine. It is an honor for Pondy Lit Fest to be the first platform where they will discuss their research.
Oh, it's working all right. Hi, good evening, everyone. This is on Alo's insistence, a purely scientific discussion that involves me. And I'm very thankful for it. There is not going to be any politics in this for those who are disappointed at the beginning itself. But as I say, please do not mix politics with science, because then all you will get is political science. So this is purely going to be scientific, and I'm absolutely honored and blessed, in fact, to have my two closest collaborators in our center with me here. One is Professor Shailja Singh, parasitologist, and the other is Professor Soumya Pati, who's a neuroscientist. And I am someone who dabbles in a lot of stuff, but I'm not able to pigeonhole myself, something who does jack of all trades. But I thought we'll give you a glimpse of in the next half an hour, because I insisted that I'll keep 15 minutes for question and answer session. So please feel free to ask any question you may like. That also would be a barometer for how closely you've been listening to us for the first half an hour. So I expect a few questions there. It's very disheartening. You say, well, we have half an hour for questions, and then you wait for 5 minutes and no hand rises up. Then you send someone, iska haath utha puchega kuch. So we thought first half an hour we'll give you a glimpse of the kind of things that we've been doing in our lab. More importantly, with the kind of budget that we have. Now, it is well known that it takes around 3/4 or $1.75 billion, let's make it $2 billion to round it off, to get any new drug into the market. $2 billion. We had $200. So thought, what can we do about it? So this is the story of what we did with the limited amount of budget that we had. Initially, what we thought we would do is, I don't think I need a pointer, but I'll just want to fall off from here, is to look at the pathogens that we're dealing with. Most of them, I beg your pardon. This one's not working. Right. So, to look at the neglected diseases and how we can combat them. Now, the most obvious one is to make a drug against it. Let's say you have a neglected disease like leishmania, malaria, tuberculosis has been neglected. Professor Pati will talk about the neurological diseases that have been neglected. Professor Singh will talk about malaria per se. But we thought one way to combat would be to look at the proteins or the enzymes, the molecular entities, so to speak, within that pathogen, that cell. So you have about 6,000 to 7,000 odd proteins. In E. coli, it's 7,000. In tuberculosis, it's 4,000 proteins. And to see if we can have a drug-like entity that would bind to that protein and incapacitate it. So if you harm that protein that belongs to that pathogen, that protein can no longer perform its function. If it is enzymatic, then it no longer can do catalysis. If it is structural, it can no longer bind to something, a host protein, to gain entry or whatever it may be. So the idea is to actually make billions and billions and trillions of small drug-like peptide entities, out of which one might go and enter inside the pathogen's protein and incapacitate it, not allow it to be active at all. That was the idea. So with $200, how do you achieve that? How do you make trillions and trillions of drug-like molecules? So we had something to go by, and that was Rubik's Cube. Now, Rubik's Cube, everyone knows what Rubik's Cube is. I think it was Hungarian inventor who invented Rubik's Cube, 3 by 3 by 3. The total number of combinations that are possible are 10 to the power of 19, out of which only one is the correct combination, where all the colors are of the same color of every face. Incidentally, I don't know about you, but I used to be really good at Rubik's Cube. I would solve it within a minute. When no one was looking, I would simply take another cube and paste the stickers from one end to the other. Unless my son caught me doing it once and said, "What the hell are you doing here?" But this is Rubik's Cube. It doesn't happen in biology that way. If you were to make 10 to the power of 19 different proteins, just one may not act drug-like. Hundreds would. And that is because people have shown theoretically that any given protein can take as many as 800 different shapes. So you have these secondary structural elements within a protein that go on to make a tertiary structure of the protein, that then goes on to make a quaternary structure of the protein. But what I'm trying to say is that every protein is endowed with these structural characteristics that give a unique identity to that protein. So you can see those levers in a key, 7 lever key, 8 lever key. So a protein can have as many as 900 levers. And depending upon those combinations that are almost endless, you end up with a unique protein or a peptide every time. So if you were to have 10 to the power of 19 different proteins or peptides in your library, surely one amongst that will be able to bind to that cavity of the pathogenic protein that you wish it to and incapacitate it. That was the idea. So now things go from bad to worse. You've got an idea what you want to do about it. How do you make this? How can we make trillions and trillions of different proteins? Well, the answer is very simple. Back of the envelope answer is, if you synthesize trillions and trillions of different genes, and you were to express those genes, i.e. translate them from DNA to RNA to protein, that's how the dogma of life goes, then every gene is going to give you a single protein or a peptide. So in order to make trillions of different proteins or peptides, all you need to do is to make trillions of different genes. And how do you make them? Well, why not we assemble trillions of different codons? Now, a biologist would understand what a codon is, a chemist would. A codon is a 3-nucleotide base. Nucleotides, there are 4 nucleotides, A, T, G, and C, that make up DNA in various combinations. And the codon, a 3-base codon, codes for 1 amino acid. And there are 20 naturally occurring amino acids. So you have a DNA, let's say, of 300 bases length. There are 100 codons there. 300 by 3 is 100. And each of those 100 codons is coding for a particular amino acid, one amongst 20. And when it codes for it, that's how the protein is made in nature. And the protein comes out, and the protein acts like what proteins do. So if you were to assemble these basic units of life, codons, trillions of them, you would end up making trillions of different genes. And then you express them and you end up making a library of trillions of different proteins. Is it as easy as that? Unfortunately, no. But the theory part is sound. Because if you look at the genetic code, you have 4 objects, as I said, 4 bases, A, T, G, and C. And you have 3 base positions of the codon where these 4 objects can go. So you have 3 chairs and 4 people can sit on it. The total number of permutations possible is 4 to the power of 3, which is 64. So in a genetic code, you have 64 different combinations of codons that give rise to not only stop codons, that give rise to 20 naturally occurring amino acids. If you look at trying to assemble, let's say, a 300 base pair long gene, you have 20 codons, obviously, each of which gives rise to 20 naturally occurring amino acids. And 300 by 3, you would have 100 codon positions. So you have 100 chairs and you have 20 people. The total number of combinations and permutations that are possible to get different genes is 20 to the power of 100. There are only 10 to the power of 86 particles in the universe. So in a test tube, you are creating theoretically more number of proteins and peptides or genes, equivalent genes, than there are particles in the universe. So surely you don't need as many as you can generate. But of course, as most good ideas, there is a stumbling block. And the stumbling block is that the melting temperature of this codon, because remember, DNA is a duplex, it's a double helix. So you have two of these base pairs that are connected, 3 base codons. So it's like a brick. And each of these bricks where it's non-covalently attached, so they're not attached together, the melting temperature where those two strands of the DNA, codon DNA duplex, come apart is actually sub-zero. So at the temperature at which you're going to ligate these codons to make this gene that you would then translate to get a protein, that temperature of the enzyme is 14 degrees. Ideal is 37 degrees, but 14 degrees, but it is above zero. And the two strands of that DNA would simply come apart. So you would never be able to join those bricks. And there are other issues as well. You have large scale mismatching and others. So we thought, well, if assembling codons is a problem, can we then assemble, instead of 3 base duplexes, double helices, can we assemble 6 base double helices? So instead of having a 3 base brick, can we construct a 6 base brick? Because then what happens is, if the melting temperature of a 3 base brick was sub-zero, the melting temperature of a 6 base brick immediately goes above. So it's about 16 to 20 degrees, which is above the ligation temperature. So you would be able to join together or ligate these 6 base bricks. But more cleverly, if the first 3 bases of the brick were exact complement of the next 3, then you only need 1 strand of that brick. When you cool it down, it's a palindrome. It will find its own and make a double-stranded helix. Like this. If there is ATGCAT, it will find its own strand and it will make a double-stranded helix, which is 6 base pair duplex. So you don't need to do anything. You don't need to find another 3 base duplex or 6 base oligonucleotide to combine it to the first 6 bases to make this dicodon. It will simply, upon cooling, make a dicodon that can then be expressed to give rise to 14 judiciously chosen dicodons. And each of these dicodons then reflect the proportion in which these naturally occurring amino acids exist in humans or in E. coli. For example, you would find, if you were to take all the codons that are used in, let's say, a bacteria E. coli, you would find that 30% of them are hydrophobic, 10% are acidic, 10% are basic, so on and so forth. So to make a library of these dicodons is as simple as simply having a vector into which these assembled genes would go in. And you would have these dicodons floating around in the test tube that each is coding for 2 amino acids. And when you add the ligase to join these bases, they would simply come together to make DNA strands of various lengths that you can control. And once these DNA strands, unique DNA strands, are made, all you need to do is to express it to get a unique protein. So in one stroke, in less than $200, you are getting a library of trillions and trillions of these peptides. Each of them is unique. This is at the amino acid level. If you were to look at the red ball, which is coupled with the white ball, if the red ball is polar and the white ball is non-polar, the way these dicodons at the protein or the peptide level come together, we have shown that they give rise to any protein that is not simply without any secondary structural elements, which is very important. There's no point making a peptide library where the peptides, those trillions of peptides, don't have any shape. It's useless. Those peptides have to have shapes. Once they do, once they have the shape, then you have a very able library that then we decided we would introduce it to a protein from a pathogen. Now, how do you introduce it? You've got this library of these different peptides, drug-like entities, out of which many might be. And on one side, you have an enzyme that you have chosen or a target you've chosen. We decided that, in order to do this, we need a system which works inside the cell. We need a system whereby we would introduce this library into a position where you have on one entity, this is just to tell you on the molecular scale what is happening. You tag your library to this red protein, which is a lambda CI. Don't need to worry about the specifics of it. You tag the pink, which is the target protein, into the blue one. And if the green entity, which is a library member, interacts with, binds to the pink entity, which is your target, it accelerates this yellow gene. It starts to produce a protein that converts, if you give a chemical compound, it changes the cell from white to blue. So if you have an interaction between a library member, out of which that library member is a drug-like molecule, and it binds to your target protein, which is what you want, you will have your bacterial colony turn from white to blue. This is what you would see. So you have a plate on which you plate out, and you have all these colonies. You would see a blue colony and you would see a white colony. The blue colony would indicate that you have found your target hit, and the white colony indicates that no, out of that library member, you haven't found anything that binds potently to your peptide. Having done that, oops, I seem to have finished my thing. I'll pass it on to Shailja Singh. But to end this part of our talk, this is just a plethora of after this, how we've used this technique to change our strategy a little. And that required changing, because if you were to target using drug-like peptide entities, proteins of pathogens, pathogens are much more cleverer than humans because they've survived longer than us. They've had to. And very quickly you find out that the pathogen changes the target protein that you're trying to target so that your drug is no longer effective, and that is called resistance or resistance. And what happens is that right from the time the first antibiotic was made, penicillin, within 5 years, you had the bacteria develop resistance to it. And we've seen now there is not a single antibiotic against which the bacteria haven't developed resistance. In fact, the last gasp antibiotic called vancomycin, there is a strain of bacteria that has developed resistance even to vancomycin. So this is one of the biggest challenges that human health is going to face in the next 15, 20 years. WHO, World Health Organization, not Wuhan Health Organization, has, although the differences have been blurred, someone has snorted that line. This has been shown to be the biggest challenge, bigger than COVID. It's going to take more lives than COVID, unfortunately, unless we tackle this. But you can't tackle it if you continue to make drugs against proteins that are pathogenic proteins, be it parasite, Plasmodium falciparum, be it Mycobacterium tuberculosis, be it methicillin resistance staph infection, MRSA, because bacteria change. They reproduce so quickly that you end up with, and that is what evolution is, you end up with an entity which your drug is no longer able to bind to. So we thought, why not we look at something else that these pathogens require in order to invade, in order to survive within a human cell, or outside but within the human body. And the reason for that is that because these bacteria and these pathogens have co-evolved with the host, i.e. humans, for millions of years, tens of millions, perhaps even a billion years, they have hijacked host machinery and host proteins for their unique use. And people have shown that HIV, for example, requires 250 human proteins to survive. Mycobacterium requires 300 human proteins to survive. People have made these lists. So these are host proteins that are absolutely essential for the invasion and the survival of pathogens. So we thought, if we target the host proteins and we make a drug against those host proteins, and those drugs work, and we incapacitate those host proteins, the bacteria or the other pathogens will die because they will not be able to use or hijack the host machinery anymore. And the beauty of it is that the bacteria are incapable of changing the host protein. So there will be no case of resistance because that's not a bacterial protein or a parasitic protein. So to cut a very long story short, we've used this technique now across species for Mycobacterium tuberculosis, for parasites, Plasmodium falciparum, which Professor Shailja Singh is going to talk about, for Leishmania, for other neglected diseases. And the principal aspect of that is that once you target a host protein, we found that there are some host proteins that are so valuable, specifically to pathogens across species, that if you were to target them, you not only stop the invasion of Mycobacterium tuberculosis, but you also stop the invasion of Plasmodium falciparum. So it's not just that bacteria have co-evolved with hosts. Bacteria have co-evolved with hosts as well as parasites, as well as other bacteria. It's almost a synergy. And I'll end that before I pass on to Shailja Singh, just to tell you, don't take it as a very macabre thought, but people get very logically so very scared of COVID. But the fact of the matter is that 12% of our genome, human genome, is actually viral DNA. We are here, we have survived only because of these viruses and bacteria and parasites. So there is a tremendous synergy and symbiosis between all living beings, and we are a reflection of that. And that is the reason why we are targeting host proteins rather than parasite proteins. Can I now move to Professor Shailja?
Thank you, Anand, for introducing me and our work together. So as he already described that how we are identifying the new inhibitors, now my job is to introduce the pathogen to all of you, the one which we are dealing with. It's malaria parasite, which causes malaria. Sometimes the malaria is too severe, and because the children's less than 5 years of the age, still lot many are dying, specifically in Africa and tropical worlds like India also. At the same time, we also work on Leishmania, where we do know dum-dum fever or the Kala Jar, that is also causing lot many and toll in human health. Apart from that, we also work because these pathogens not only causes some pathogenesis, but they also introduce some kind of mental health problems, because these parasites travel to the brain and then you got some disorders. So in that case, Dr. Soumyapati is going to talk about how these pathogenesis, how these infection can cause some disorder in the brain and how we have to tackle them. So in that case, like first thing, as I said, our approach is to target the host rather than the pathogen, because we want to control, keep the control with us. In the sense, whatever drug we are designing, we want that should just work. Parasite, whatever it is clever enough, but it cannot change. So in that case, keeping that in mind, first of all, like here, I am going to show a movie where you are going to really admire this, because here you can see the human red blood cell, which is just flowing in your body. And then the one which looks in the green, that means in that cell, you have a parasite, that is Plasmodium falciparum, which is a very lethal form of a parasite which causes the cerebral malaria. And then you'll see how clever these pathogens are, how they are coming out by busting the human red blood cell, again going to the new red blood cell, interacting with them, how they are invading into that host red blood cell. So what do you want to do? You just want to stop the exit from one cell, and then they become more in number. You also want to stop the invasion to the new cells. How are you going to block this? That's what, here comes the collaboration. What he has discussed, that the interaction of two proteins mediate this interaction of host and the parasite. So you can block the interaction, that means you will block the invasion process. Or you can block the hijacking of the machinery by the parasite, you will block the exit of the parasite. Basically what we are doing, we are just doing locking of the parasite inside the red blood cell, or we are not allowing the parasite to go inside the new red blood cell. Just to, you will really appreciate this, because it took me almost 6 months to achieve this video, where I just mimic everything what is happening in the body under the microscope. So we took the human red blood cell, we infected them with the Plasmodium falciparum, and hours together I was just trying to observe the phenotype which really we feel. You all must be knowing, when we get fever, we get the chills. Why? Because that time your red blood cells are just blasting. And then the parasites are coming out in numbers and they are attacking to the new red blood cell. So that is the pathology associated with the malaria. You are getting fever as well as you are getting chills. So you can see here now. Here is the blast. Many parasites came, and in the target red blood cell they entered. And then you can see not only when they are entering, they are also hijacking the host. There is a process called echinocytosis. The host is trying to accommodate the parasite, and once the invasion has happened, the host has also tried to become the same. What do you want to do? That's what I said. Just block it. Either don't allow them to come out, or you don't allow them to invade to the new red blood cell, which I showed. And the same thing, when it is happening, the parasite is also releasing some of the proteins which are really dangerous. Because if we all know that when we get infected by malaria, we also get anemia. Why? Because first of all, as I said, the red blood cells are rupturing, and some parasite factor which are being released in the blood, they are also attacking to new red blood cell, and your red blood cells are dying. In that case, the hemoglobins are out, you are getting many pathogenesis. Now, you just don't want to lock the parasite, but you also want to protect your human red blood cell. So you need to know which are the factors, which are the proteins which are doing so. Then go back to Anand's strategy, or some natural compounds, or some chemists. Give me some compound which should block this protein, so this protein should not attack to the new red blood cell. And it should not lyse the red blood cell, so we should not get anemia when I'm getting infection by malaria. So in that case, this is a human brain cells which we just took, because as I said, parasite travels to the brain. It is releasing some factor when it is coming out from the red blood cell. It is interacting to brain endothelial cell, and your brain endothelial cells are dying. That's what all the brain disorders are happening when we get the cerebral malaria. So here you can see how these cells are responding. Very nice cell which is there. Again, it took a lot of time to capture this. This time we made the protein which is coming from the human malaria parasite, and we allowed them to interact to the host, and you can see how it is causing the damage. The brain endothelial cells are just going, all abrupt process are happening. There is a blebbing. Then your cells are now not in a normal morphology. They are just dying. Apoptosis is happening. A special kind of a death, because even the death is so systematic. It's just not the cells will die. It is also going to follow the process. And the process, if you know, you can block the process of death of your human brain endothelial cells. And basically, you're trying to protect it. So you can see this, how the cells has become. At the same time, some molecular level, like how the calcium, those are the very detail which we, as a scientist, we do because we want to block the processes. And the next thing, as I said, not only the brain endothelial cell, but your human red blood cells, you can see, like it's a really very interesting for me, because we just took my blood, this is my blood, where I put the parasite protein. And you can see the cells, because the calcium is going high, all are now green, then they become so swollen, and then suddenly they are like just rupturing. And this might be happening when we are getting the human malaria parasite. So with all this information, which took us a lot time, meaning 6 to 8 years, of course, but at the same time, when we know which is causing this all trouble to our cells, we can block that by designing some small molecule or some peptides or some natural compounds, because those are the good ones already tried and tested in traditional medicine. Why can't we give some scientific rationale to those natural compounds which can protect our normal cells, and at the same time they should kill the parasite by blocking the host factors which are supporting the parasite growth. And for doing so, like we could show many like not many publication. I'm so happy and fortunate to be such a big group I'm heading with, along with Anand, and we are really doing wonders in case of malaria, tuberculosis, and leishmania. We could design many compounds. This part of the story I just wanted to tell you, like artemisinin is the only drug available for Plasmodium falciparum, but at a modern medicine approach. We know how this medicine works, so we could make some iron nanoparticle as a nanomedicine approach. Then we went back and we could design some, we could know that the Ayurveda has law of Bhasma. By mixing the two, which is we could show, and this is also a published work, that not only we can design a new drug, but we can also make drug which is already existing more efficacious, because it's a fortification of some component from our Ayurveda to make them more effective. And then also some interaction with the Nigerian country, because there we know that the malaria is too much. There are some already Ayurvedic some plant-derived compounds they are using, but we can give them the scientific rationale why it is working, which protein it is targeting to. So those are the things. And then not only identifying some of the natural compound, but we are also synthesizing some natural compound by guided by those are present in some plant. Those plants are being used for parasite or for malaria fever, for leishmania. Now we can make in the lab, and if we can make them, we are really going to use it for the as a drug. And at the same time, we also are collaborating with the other scientists to have a catalog from our India, all natural compounds which are there in the plants which are being used for the malaria treatment, leishmania. And in that case, we can have a catalog, and from that catalog, one can pick, like Ayush are also helping us to do so, and that will be anti-malarial in action. And finally, just because I belong to Chhattisgarh, and there I could know in Bastar district, the malaria fever patients were being bitten by some ant, and those was the normal treatment by the tribals, and they were doing, and malaria was cured, but nobody knows that why this therapy is working. So I could give some scientific rationale to it. I got the ant, fire ant. I could analyze what is there in the venom of the fire ant, and then tested in the lab to show these ant venoms are now why they are anti-malarial in nature, because I could identify some of the drug from that. And that was it. And finally, I will ask Dr. Soumya to take us through how these pathogens, how they interact to the host, to the brain, and what kind of nonsense they can cause, and then how we can protect.
Hello, good evening to everyone, and thank you, Shailja and Anand, to give me this platform to share my knowledge with you. By profession, I'm a translational neurobiologist, and as both of them have already introduced to you that how these pathogens can impact the system, I would be talking about a very different aspect, but of course that how this virus, pathogens like parasites and bacteria can impact the brain. So just to introduce to you that my work is, which I have shown here, I'll be talking about brain in a dish. So in comparison to other organs in the body, brain is something is we cannot touch. We cannot have any biological sample to test. Even if somebody is infected with some kind of virus or pathogens or any tropical infection, we cannot just go to a clinic and say, you open my brain and have some samples. So I'll be exactly taking you through a short journey to understand how brain functions. Just to introduce you, that brain is something like a supercomputer device, so which runs trillions of algorithms at a single time. Even I'm talking to you, my brain is involved in multiple processing of the image, because I'm also while talking to you, I'm also thinking what will be the next sentence, right? So I repeat that sentence inside my head also. All these functions, which we call cognition in scientific term, it's controlled very heavily by algorithms which are people now exploring. Nobody has known how it works. So imagine a supercomputational device which is wrapped by biological components, fueled by chemical moieties or chemistry. Is very interesting. Now, these pathogens and viruses and fungi or toxins, bacterial toxins, any tropical infection which anyone gets, this can surpass the blood-brain barrier, we call blood-brain barrier, and they can easily go to brain. So I am going to show you some of my very good developments from discoveries from the lab, and also I work with these two very intelligent scientists who are very good friends and collaborators also. So we work in a lot of drug discoveries, and which targets the infection in the brain. So while I introduce you to the impact of these pathogens in the brain, which I call the infections, which can challenge the immune system. So there are two things we look when the person is infected with any kind of infection is metabolism. So I'm giving you an example of one of the neurological disease which I'm going to describe to you is called autism spectrum disorder, which is very heterogeneous. Nobody knows why it happens. So it happens to kids at the age of 2. And the data says, or the clinical information says, that the mother, when is infected with even a small flu or any virus or any kind of pathogens, it challenges its own immune system, and the fetal brain, the developing fetal brain, is directly get involved and damaged, right? So I am giving you example of Takshila University, which was developed in 700 BC. Why I'm saying? Because most of the scriptures, like written by Charak, who was also father of the Ayurveda, he initially at that time gave the metabolic products, or the metabolism can be a windows to detect any infection in the brain or in the body. So a lot of his scriptures, which is like Charak Samhita, and if you can happen to get the glimpse of it or some portion of it, you find that he talked about Agni, which is kind of a combustion of the food which we take, and the metabolic impact or changes, how it impact the whole brain and brain activities. So so this was developed in 700 BC, and the metabolic study came later on. I don't know how. Sorry, I'm not very comfortable with Apple, but maybe Shailja can say. It's working. Oh, yeah, maybe. I'll open it again. So, okay, next. Yeah. So after this discovery, much later in 1506, there was a work by Ulrich Pinder, who is a scientist who actually showed that the smell and color of the urine can show you the change in the highly metabolic rate of your body. So what I bringing you about the neurodevelopmental defects which are generated by this infection in the mother, and during the early period post conception stages, how it can change. So I'm giving you example of a kid who came to my lab, is 4.5 years. She developed autism spectrum disorder, but there was no as such diagnostic tool. As I told you, there is no biological sample. And she came to my lab, and within 6 months, she has a speech loss, hearing loss, and the muscle tonicity or the strength gone. So she's partially blind, 80 to 90% visibility remains. So the father and mother was holding her, that they came from USA just to find out a hope in India, maybe Ayurveda or something, some supplement can save them, because there is no treatment as such. So what we do exactly to study any infection or the infection induced damage to brain, we generate the brain cells from the dish. So how do we do that? We look at the patient information. For example, this kid is having severely speech and motor defect. Motor means the movement defect. So we know which area of the brain is involved in what kind of activity. So we do the genome sequencing, means we need to find out if it has any mutation is linked with that. And then based on the changes of the behavior, we generate the brain cells from the skin cells. We call pluripotent stem cells, or there is a way to generate embryo-like stem cells from the skin. So then, I'm just giving an example. This is called, we basically mimic something called mini brain in the dish. So I'm giving you example from, we cannot remake the embryo of the child which I showed. So we take the skin cell and we generate the mini brains, because that is the way you can target any drug or any of these drugs which is, Shailja was saying, which can work in cerebral malaria or any pathogenesis, where they will try. You cannot directly inject that drug into the brain because you don't know the impact. So we generate a patient-derived stem cells and the patient-derived neurons to see if it is exactly working. So these are some of the good work from the lab. We have generated the patient stem cells. I'm showing you what we might be knowing called neurons are the firing wires in the brain, which contributes to all the cognitive abilities. Even, we know that all cognition, like the intellectual ability to do any physical activity, also controlled by brain. So these are the cells which are basically patient neurons, which we grew in the dish itself in the lab. And these are being targeted by different drug-like molecules, which we discover from our collaborators, and we test how it impacts. I am giving you a very good example of one of the antidote which we developed to target these pathogens. And this is very, very simple molecule, glucose-derived molecule, which we tried in animal model. And you can see the animal which are not having their died, but those have this drug, simple glucose-derived molecule, which is absolutely non-toxic because the backbone is glucose only. It is showing high survival. So this drug actually is given to target the toxins or infections which are developing in the early stage of pregnancy, because we don't know what is going inside that type. So this is what we studied in the animal model. Now we are having our own patient-derived stem cells or neurons, which we are trying. So this is just a glimpse of how the drug discovery from the bench what we got, we can translate. We call it as translational medicine, or we call it bed to bench to bedside. So we took a challenge from a bed, we bring into bench, we develop the idea to generate the model to study them directly from the patients, because we cannot open the brain. And then we get information that this kind of target can have an impact on the metabolism or the infection directly, and then we translate to bedside. So this trilogy is bed to bench to bedside. With this, I'm just closing my talk. So basically, both of my collaborators told me, you don't show much data. So I have I have squeezed it into a very small story. So basically, to grow these human neurons, it takes 6 months because any functional live wire in your brain, it takes 6 months. So we all are having neurons, and we are all having newborn neurons every day, up to 600, but most of them die. So I have we have a PhD student in my lab who got stroke while working on the bench. And we saw developing the neuroprogressive damage. By 24 hours, part of the body was paralyzed. Even then we were working, we cannot stop that. But then later on, we found out that because that we thought of giving everything, giving the stem cells to regenerate, but it is very difficult to generate a network. You can generate live wires, but generating a network is difficult. So we do multiple ways to have information where we can target the disease through a model. So that's how I just want to close my topic. Thank you very much.
For whom the bell tolls, how many minutes do we have for? Very kind of you. Glory to questions, please. Yeah.
My question is to Shailja Singh. You mentioned the fire ant and the low bhasma.
Yes.
So there is a lot more in the Ayurveda,
Yes.
which we need to.
Yeah. So, yeah, I totally agree, and that's what we are starting a project with Ayush Ministry, where, as you said, ashwagandha, and there are many plants which are already been shown to have anti-malarial activity, but nobody knows, because in a plant extract or any Ayurvedic medicine, first of all, the problem is we don't know exact dose of that compound present in the Ayurvedic medicine. So if as a scientist I can give the rationale, this much compound of that nature is there in that extract which we are using, though it's immunomodulatory effect or anti-malarial, anti-leishmanial or anything. So for that we need to do research, and for that only, as I said, the scientific rationale is very much necessary before using any compound. For example, giloy already been shown, this is saving immunomodulatory effect, but which component of the giloy? I need to know what is the dose which should be taken in the form of extract, and that's what I say. But in this case, I'm really proud to have this work done, where I could combine the law bhasma with artemisinin, and that's what we got the Gandhian Innovation Award from conferred by President of India, where we could show this is a very innovative approach where we can improve the efficacy of known drug by 10 times. As I said, the problem is the resistance, and if I can improve the efficacy without having a much effort of designing new compound, it's always good, and I can take the help from Ayurveda medicine. Yes.
I think it was Shailja that we mentioned that you take 6 to 8 years to complete a process of investigation.
Yeah, more than that, actually 22 years of my research career.
So how do you benchmark each particular period that you are on the right path? Because it almost, to me, it sounds like as if you're working blind. I'm sure you're not working blind.
No, not at all. I'm not blind at all. Though I don't have even the specs, it's just for some reading. So I really, I'm very focused in terms of my journey to research career is from the day one. I was very much aware what to take and what not to take. For example, understanding a disease is almost very much important. Having the basic science information. If you don't understand the pathogen, you don't understand the disease pathogenesis, how are you going to treat? Then go after that, you are going to dissect it more, that which are the proteins which are responsible for causing this kind of a damage to your cells. And then which kind of a damage? If at all it is killing your cells or killing the interaction of your cells, those kind. So from the after my PhD, during my PhD I work on cancer. Again, it's a drug discovery. Then I could turn all my knowledge from the cancer biology towards malaria, because I think being in India and that to a tropical world is really facing a lot of problem. And malaria, these are the neglected disease. Nobody bothers about it. So in that case, we need to understand the pathogen. And I think I'm really happy to say that mentors, they played a big role, not to be defocused, not to be blind, just follow the right scientific path to give the answer, to give the rationality to the all scientific world, that yes, we can give the information which is hidden in the host or in the parasite. Yeah. Thank you.
Hi, Anand. This is Naveen. I have this question. I'm actually a microbiologist, my basic graduation. So 1990s, I used to visit AstraZeneca and they were doing this research on malaria. So they were the pioneers. So I'm just wondering, is it I'm time struck or time traveled, and 2023 still we are looking at, is there any evolution happened in the whatever the host or organ, whatever is there, or is there proper funding is not there, or is it really neglected because only it's happening in tropical countries and African countries? So what is happening? So this is my first question.
This is fantastic. In fact, let me tie up with a comment which I could make on Ashokji's question, because I think the two are connected. When he talked about the time frame and you talked about the development that usually happens and is there something happening. So science is a very esoteric thing. I mean, if you look at the genesis of science itself, especially experimental science, back of the garage thing 200, 300 years ago, it's a very personal thing. It's almost like a hobby that then developed onto something. So there is passion involved. But at the same time, earlier when passions used to be involved, there was no time frame. Now we can't survive like that because everything is time bound and with grants and everything. There are three things that come into this. One is, if you end up with your discovery or invention to negate an existing dogma, then it is very difficult for you to win or have victory, at least in the short term, because so much has been invested not only by academia but by the pharma industry into that dogma. So many billions are being made by the existing principles, either fundamental or applied, that if an invention comes and says, well, this is no longer true, you will find it incomprehensibly hard to overturn it. And one of the, it's not just me saying it. In fact, S. Chandrasekhar of the famous Chandrasekhar limit, he gave that idea in the 1930s. He got the Nobel Prize for it in the 1980s because Sir Eddington said this is a rubbish idea. And in fact, it was Max Planck, famous Max Planck, who said, science proceeds funeral by funeral. So when a person, a very famous person who's given an idea, when that fellow dies, the new idea, the correct one, can come and take its place. Very beautifully said. But one glorious example was, I think, Barry Marshall, who is perhaps the only scientist to have not only got the Nobel Prize, but also the Ig Nobel Prize. Now, Nobel Prizes, we all know, are given to tremendous discoveries, innovations, as well as to fact checkers, but that's another matter. Ig Nobel Prizes are the ones who are given to research that are carried out by scientists that are completely whimsical and, over the top investigations that are so zany and arcane that you say, my God, is he actually, in fact, has he done this experiment? How amazing. This is what the beauty of science is. So that's the Ig Nobel Prizes. He's the one who's got an Ig Nobel, as well as, so for example, the last Indian who got the Ig Nobel Prize, he published calculating the surface area of an elephant. Things of that sort, and that is what makes science so beautiful, because you get an idea, you want to do it, you pursue it. And he went to Kerala and he had to experimentally determine the surface area of 17 elephants to get an average of what he did and, how he survived. But so Barry Marshall said, The dogma till the 80s was that you get ulcers because of proton pumps, and there is acid reflux, and all you need to do is to take proton pump inhibitors, ranitidine being one example, and so many others, and you would cure your ulcers. And that dogma lasted for about 40 years, and these anti-ulcer drugs or proton pump inhibitors were the world's largest selling drugs for 30 years, until this Australian guy comes along and says, look, rubbish, ulcers are caused by bacteria. And the pharma industry said, are you mad? How can a bacteria survive in your stomach which has a pH of 1? It's nonsense. He was denied grants. He was almost sacked. He went to another institute. He was refused funding. He could not work there. In frustration, he took that bacteria and he drank it. And he got ulcers, and then he took antibiotics and he cured it. And he got the Nobel Prize for it. H. pylori is the thing. It's just remarkable. So when you talk of time limits, sir, I don't think there is any time limits to science. It's just one of those most beautiful things that are purely driven by passion. And, life, its existence, money didn't matter to Barry Marshall, doesn't matter to Soumya, doesn't matter to Shailja. Yes. You could drink it in secret without telling anyone, but and also the problem is many antibiotics you might take might not work, might be spurious antibiotics, so you might actually die. Now let me come very briefly to the other question that you asked. And there I would say that, and I've said this before, more than India being a land of opportunity, I think India is a land of missed opportunity. Let me illustrate it with an example. Shailja talked about artemisinin, making it more powerful. But the fact of the matter is artemisinin is now being used. It's perhaps the only viable anti-malarial. Before that chloroquine, there is almost widespread universal resistance by the parasites to chloroquine. So people only give artemisinin. In East Asia, there is resistance against artemisinin that's increasing. It's coming to India. There's a lot of investigation in that. How many of you know of Lakshmi Kutti? Could you raise your hands? All right. One hand has gone up. Lakshmi Kutti is an 83-year-old frail woman who lives in the jungles of Karnataka and Kerala. She got the Padma Shri because she's worked for 60 years of her life in very carefully investigating the medicinal plants of Western Ghats and making a library, cornucopia. Her library is 500 plants strong. She's been curing the tribals who visited her. People know that chloroquine is a quinine is the result of a bark of a tree. And Peruvians had been curing malaria for 1,000 years before Wellcome came along and packaged it in a tablet that could be eaten, and he made billions out of it. But artemisinin, the woman who discovered it is a Chinese woman. Was that accidental, or did you just pick up the bell, or okay, just 30 seconds. Inverted, yeah, just about to finish. And she, from a plant in China, she isolated artemisinin, and scientists nowadays vie for publishing something in a journal with an impact factor of 60, 70, 80. She published in a journal with an impact factor of not even 1. She got the Nobel Prize for it, artemisinin, very recently, 5 years ago. The problem is we don't even have an institute in India that could take, forget about 10 or 50 or 500 of Lakshmi Kutti plants and isolate the ingredient. We don't even have one who could do that. There should be 500 Lakshmi Kutti institutes of natural products, isolating the active ingredient of each one of those medicinal plants. Who knows? One of those molecules might be a blockbuster anti-diabetes drug, anti-malarial, anti-tubercular. But you know what will happen? That some scientist or a bunch or a lab in Nordic countries in Sweden would come to the Western Ghats, take a couple of Lakshmi Kutti's plants, take them back, isolate the active ingredient, find out that it is a better anti-malarial than artemisinin or a better anti-tubercular drug than rifampicin, get the Nobel Prize for it. And then satisfied, we would give Lakshmi Kutti the higher Padma or Padma Bhushan. Why can't we do it? If a Chinese woman can isolate artemisinin from a plant, we have 500 plants waiting. So to answer, I hope that answers your question. There is not just patience that is required. It is the support that is required for doing science. Science requires as much patronage as classical music does or the arts do. The day people realize it, I think we would rise ourselves a notch higher. Thank you. Can I, with the permission of the chair, because she's answered a question, I've answered a question, is there any question relating to the brain?
You talked of disease, you talked of autism that affects children when mothers are infected during earlier in their stages of pregnancy. So is it a better way to have more awareness that these are innocuous diseases that mothers have, and they should actually be careful during pregnancy rather than treat it when it becomes a neurological problem in a child?
That's a very good concern and good question, actually. So, in India, actually, this awareness is not there because they generalize all neurological disease kind of same, you know. But in European countries and other countries, they do have very good clinical data which says that there are multiple factors which can affect. Briefly, any infection during maternal post-conception period. Stress is one of the major factor. Dietary habits, over-vaccination, and parental age. All these factors matter. Now, in India, to reach to grassroots level awareness to the women who are conceived is very difficult. But Indian government is started some kind of awareness programs where they are directly, like for me now, the patients, they search because Google is there anyway. They go through certain intermediate coordinators in the village or in the dispensaries who guide them to connect to the clinician scientists, to talk to them to find out some resolution. At least now we have started spreading kind of awareness to the women who are pregnant to take care of certain factors which should not impact the fetal brain. Like stress is because it's a nowadays nuclear family. Earlier it was not like that. So stress is one of the major factors which Indian, the young population is not really exposed to. They, they don't know how to handle the stress. So maternal stress is very damaging to the fetal brain. This is one thing. So we are trying to coordinate this to whomever is the intermediate person who can translate the knowledge that these are the factors like fooding habits. It should not be stored food. Any stored food contains bacteria. So bacteria, to survive, they produce toxin. Toxin has a beautiful capacity to cross the blood-brain barrier, as Shailja is showing. So that earlier damage, what is happening in the fetal brain, you cannot know till the child is 2 years age. By the time it is 2, you see their perception is low. They don't understand the command. Whatever you say, they don't have a perception. We know something which is sweet or sour because we have a perception in the brain and we have a database for that. The child do not have any such kind of perception. So it is very difficult to train a autism. The child is who is suffering from ASD. So now Indian government has probably awakened and it is giving more funding to this kind of awareness program. So hopefully something will be done. So by the time we can keep on doing whatever we have to.
So you said stored food is a problem. So Mysore Pak is a